Welcome to the Lower LA River. Let's introduce some of our stream team members and our testing sites.
Sarah Kay, Riverfront Park.
Monet Pedruzini, Hollydale, and Rio Hondo.
Melissa Zelaya, Compton Creek and Confluence.
Sarah Valencia, Willow.
And Jasmine. While we get ready to take samples in the LA River, we make sure to plan who will be going into the water and who will be on the sidelines, not in the water, as a safety protocol to make sure we have someone ready to respond to any injuries that may occur. While we are being safe, we are also enjoying the beautiful living ecosystem the LA River has to offer. We observe surrounding flora and take note of the presence of algae. We also have encountered a turtle, many birds, a few dogs, dragonflies, and horses. We see lots of families walking along the bike path, bicyclists, walkers, and joggers. We witness the good, we also see the bad. Lots of trash and debris left behind, such as a hanger, tire, and cart.
With the water thermometer on our boot, we carefully make our way into the stream, noting water clarity, color, and odor. The first thing we do is get turbidity samples, which is how much stuff is in the water. We fill up 2 small vials with river water, first rinsing each vial. Once the samples are collected, we move on to the river water sample that we will be testing for bacteria. We label each sample with the date, site code name, and time. Then we scoop up our sample and get it on ice right away to prevent any bacterial growth until we are ready. Some sites have digital temperature readers installed, taking measurements every 30 minutes. This gives us more vital information that we can download and email. The next step is measuring turbidity. We wipe each vial and place it in the calibrated reader to see how many particles are in the water. Then it's on to the next site.
Meanwhile, back at the lab, safety first. We must first gear up with goggles and gloves. Then we proceed by sanitizing the lab benches to prevent any cross-contamination. Our samples, tests, disposable pipettes, and sterile buffers are set up neatly, consistently, and by chronological order. This is a lab blank sample we take in the lab with distilled water that we process the same way as the others for quality assurance. One buffer will test for E. coli and coliform, while the other tests for Enterococcus. Each buffer container will receive 10 milliliters of the sample. Enterella and cholera is the nutrient that the E. coli and Enterococcus bacteria will eat. We mix the buffers until fully dissolved and put them into trays containing small and large wells. These trays are properly labeled with the appropriate sample information. Labeling our trays ensures we know exactly the location, date, and time of the samples. Now we put the trays in the incubator so that the bacteria has the perfect environment to grow for 24 hours. All containers used get rinsed and bleached to prevent any harmful bacteria from contaminating other surfaces. And that's all for today. See you in 24 hours when we take out the trays.
The next day, we take the samples out of the incubator, noting the time and date removed. The test for total coliform is the easiest to determine, as any wells that are yellow are positive for that bacteria. Our lab blank is used as a control. Next up, we test for E. coli and Enterococcus. We place samples under UV light to check for fluorescence, which indicates a positive result. Brighter wells are easy to read as positives, but sometimes they can be much harder to determine. We form a consensus among several of us, using prior readings as a guide to determine a positive versus negative result. Once the analysis of our sample is done, we then log the data both on paper and the computer. The final step is to update the river report card. Unfortunately, with the samples we have gathered thus far, all of the southern river sites are under a red grade. This means that the level of bacteria detected exceeds safe levels.
It's Heal the Bay's mission to report this data in order to protect and inform the public on how to preserve the LA River. We work to raise awareness about pollution and do the actual dirty work ourselves with volunteers at annual coastal cleanups. We see a future that preserves the natural systems of the LA River that benefit both people and the ecosystem. Our president, Shelley Luce, put it best.
We want to see nature-based projects that clean and capture water using plants and soils, that also green and cool our neighborhoods.
So join us in our mission to protect the environment. Thank you for watching.